Okay, we'll wait 5 more minutes if anybody joins, and then we'll start the session.
सेफ साइड पहली बार तो ऐसे ही करते हैं, दो कर रहा हूँ ठीक है। अब मैडम मैं चला जाऊँगा बीच में, ठीक है।
Okay. So in the last classes we have read about the antigen, the foreign antigen, how when it infects our body the antibodies are generated. There is innate immunity, there is adaptive immunity, and there also we have B cell and T cells. And when there is a B cell mediated response, we have seen the generation of antibodies which will work as a memory, and when again the antigen increases, the memory cells will produce more and more antibodies that will react to that antigen and end that antigen. So today's class we will study about some tools and techniques that people usually use in the immunological studies, and that tools and techniques usually uh, employ the antigen and antibody interaction. Okay, and so let's so this is an antibody molecule. If I consider, okay, an antibody molecule which usually binds to the antigen here in the variable region. Okay, and this detection or interaction is employed in further techniques. Suppose if uh, you want to know if any particular person is infected with certain kind of pathogen. So suppose you have a set of antibodies, okay, um, or not antibodies, okay. So suppose you encounter an patient which is infected with some disease. Now you want to find out which kind of infectious disease, okay. So suppose if that person is having a malaria or a kala azar or any other kind of disease, which disease the person is having. So there are certain tests people scientists or doctors use, okay. So one of that test is recognition of the antigen, okay. So if the person is infected with that any particular antigen. Suppose a person is infected with plasmodium, it is having malaria, it is infected with plasmodium. So I will detect first whether that person is having plasmodium antigen in their blood or not, okay. So antigen is generally the protein, so it is having the parasite protein or not. So that detection I can use if I have the antibodies which can interact with that antigen, okay, or with the parasite or with the protein, okay. So in order to do that, we are supposed to have certain set of antibodies. So suppose uh, so other way what I can say is you can have the antigen with you and then detect whether that person is able to produce the antibodies or not. So suppose I'm infected with this malaria disease, and against that disease I have generated antibodies in my body. So my blood contains antibodies against that disease. So I'll as a doctor, I'll take out the patient's blood and separate the serum, and I'll detect whether that serum is having the antibodies against that specific disease or not. So in order to do that, I should have the antigen with me, okay. So antigen is a kind of protein, nothing but a protein. So you should have the antigen with you in your hand. So how you can get the antigen? So antigen usually or protein production you can do in a laboratory, okay. So suppose any particular against any particular parasite or any other organism or any other virus, okay. So against that, whether for that virus specific or that antigen or that parasite specific protein you should have with you, okay. So suppose this is a vector I'm drawing. So this is a molecular biology technique. So what we do is, okay, so okay, so let's assume you have you want to detect whether a person is infected with a parasite disease or not. So what you need parasite protein, a specific protein. So how do you get a parasite protein? So for to get a parasite protein, what you have, like some labs they culture these parasites, okay, or if any other organism. So anything from the nature you get that particular organism with you, okay, and from that particular organism you isolate the genomic content, okay. The genomic content you isolate, and there are certain, we have talked about epitopes, okay. So epitopes is the region against which the antibodies are generated, or antibody can recognize the epitope in a protein or in an antigen. So that particular epitope you need to so that they can interact with the antibody. So those epitopes from which genetic region or genomic region it expresses, those epitopes you need to have with you. So from all the genomic content that you have got from that particular organism, you amplify only that particular genetic information that codes for that epitope. Okay, so in order to do that, you utilize the molecular biology techniques. For example, first step will be you do a PCR, okay, a polymerase chain reaction. So from all the genomic content, you amplify the gene that codes for that specific pathogen or epitope. Okay, after that you get the gene. Then there are certain vectors, okay, or plasmids that are what you say that overexpression plasmids. Okay, so what if you can put that your gene of interest or that gene which codes for that epitope inside that plasmid or in frame with that plasmid, you can produce protein from that particular plasmid. You put that entire construct in a surrogate system or in a bacterial system where they can produce that genetic information, that from that gene they can make protein out of it. And then that you get the protein, purify that protein, and then that purified protein you can use as an antigen. Okay, yeah. So that is a lot a step by step process to purify an antigen from an organism. So whatever an organism antigen you need to have genetic material from that organism. Then from that genetic material you amplify the genetic region which codes for the epitope, particular epitope. You clone it in a expression vector. From that expression vector you express the protein from that gene, and then that expressed protein you purify it, and that purified protein is your antigen now. Okay, and that antigen, when you allow the serum to bind with that antigen, or to add the serum in that antigen, the patient serum in that antigen. So if the patient is infected with that disease caused by that organism, it must have produced the antibodies, and that antibodies will bind to that antigen. Okay, and then antigen antibody reaction will give you a signal that you will be able to detect and can say that yes, this particular patient is positive or infected with this particular organism. Okay, or if that serum does not contain the antibody, there will be no interaction and there will be no signal, and that says that okay, so maybe this particular patient is not infected with this particular organism. Some other antigen you use and allow your serum to bind, if that gives a signal. So that is how you can screen. Okay, so this antigen antibody reaction is highly employed to study the disease patterns. Okay, so for example, there is this particular reaction, or you can say technique, which is a immunodiffusion technique. Okay, so this is also based on antigen antibody precipitation. So what is this? There is a protein antigen and then there is a antibody. Both together, what they do, they interact. Okay, and when they interact, they make a visible precipitation. Okay, uh, and that visible precipitin is a whitish in color that you can see. So that is a signal. Okay, so antigen antibody together, they give you a signal, okay, that you have to detect. So for example, here you can see, so antigen antibody, they both diffuse towards each other. So this is a agar plate. Okay, this is a petri plate or a slide where you have made an agar. Okay, and in that agar you made these holes. Okay, these holes are there. Okay, and in between what you do, suppose, so they both are proteins, antigen antibody both are proteins. So in the middle one, suppose you have added your antibody. Okay, and in all the other one you have the what to say this antigen. So suppose this one, these two you see, this is where the antigen is present, this is the antibody is present, so they both diffuse towards each other through this agar. Okay, and when there is an altogether both antigen antibody concentration is equal, all the antibodies are occupied with the antigen, there you'll see a line of precipitation. Okay, you'll see a precipitate. Okay, so there are some areas where suppose like this one here, only few antibodies are there and lots of antigens are there, so here there would be a precipitate kind of thing. In the outer one you have lots of antibodies, no antigen, no precipitation there. But where you have the optimum or equal concentration, where all the antibodies are occupied with the epitopes, antigen epitopes, and there they'll make a precipitate, and this is directly proportional to the antigen concentration. Okay, so this is the antigen concentration and this is the diameter of the precipitating disk. Okay, this we are saying is a precipitating disk. Okay, so here this this one you can see the diameter is increasing as the antigen concentration is increasing. Okay, more antigen you will give, more precipitation you will see. Okay, so this is what, this is a radial immunodiffusion. Okay, where you can see more the antigen concentration, more bigger the ring. Okay, so this is what, and here in this case, so this is also a similar kind of set. Okay, so here in center well, in the center of this agar well, you have added your antibody. Then there are several other wells. So in each well you add different antigens. Suppose here you have added antigen 1, in this one you have added antigen 2, in third one you have added antigen 3, then 4, then antigen 5, and this is a control reaction, you have not added anything maybe. Okay, so then you allow the diffusion of these reactants. Okay, they both diffuse to antigen and antibody, they both diffuse towards each other. You may get 3 kinds of patterns. Okay, so the pattern of precipitating lines reveal the relation between the antigens. Okay, so here you see, so in the center one, this is a one portion, suppose this one portion I have taken. Okay, consider this portion you are seeing there. Here, so here you can see here the this center one, the bottom one, here it is containing the antibody, these two they are containing the antigen, two different antigens. Okay, so this one and this one they'll react with each other and they'll make a line of precipitation. This one they will react with each other and they'll also make a line of what is a precipitation. Okay, but here you can see this line, precipitation line is made in a such a way that they intersect each other. Okay, so here is a cross is happening, so this intersect, it tells about the antigen similarity with each other. Okay, so here these the lines cross each other, and then it means that two antigens share no common epitopes. Okay, so suppose this one and this one, they are making one reaction, they are doing one reaction, and this one they are doing another reaction, there is no similarity between them, that's why they cross with each other. Okay, there is no mixing of these two. If they both were same, for example in this case they both are same antigen, so they'll come in such a way that they make this arc kind of structure. Okay, because they both are same antigen, but if they are not, then it will make an independent reaction from this side, one independent reaction happening, this side there's also independent reaction is happening, so they are making independent lines with each other that are crossing each other. However, in this case, if they are both similar antigen, same antigen, identical antigen, or you can say they have similar epitope, then there is a arc which is able to form. Okay, because they both are similar, and this, if they are similar, it is kind of a radial thing, like this one, it's a radial appearance which is trying to be made here, and that is why you see a arc there. However, if there is also this pattern which is partial identity, so in partial identity lines merge partly but a spur is same, okay, which indicates the antigen shares some common epitopes but one antigen have additional and unique epitopes, so the spur points towards the simpler antigen. So this one is a simpler antigen and this one which is having additional epitope, that is why it has additional reaction which is going on, okay. So these are three kinds of pattern or precipitation lines you get. Based on that you can say the similarity or the, you can say, the connection between two antigens, okay. So for example here this also it has been seen, so this is the real image. So here you can see here this anti-serum, the serum is added here. You can see four different wells to add the antigen. This one is giving arc where both the antigens are same, okay, the arc is given. And this is trypsin, this is proteinase K, it is not interacting with this and these two, and that is why there is no precipitation line also occurring, okay. So this is a simple immunodiffusion technique which can tell whether the antigen antibody will react with each other or not, okay. The next technique that we have is this western blot technique, okay. Anything you want to ask about this one, the immunodiffusion one?
Ma'am,
Yes.
I have a confusion in between the means when there is two different antigens, they are making interaction, but they are crossing.
Yes.
But how?
That means, so suppose here and here they both are coming towards each other, okay, they both are coming towards each other. So here there are certain set of this, okay, these are coming, they both coming towards each other, okay. So what is happening here, from this side also another antigen and antibody they are coming towards each other, but here the antigen pattern is different, okay. Here suppose you will get, here these antigens are there, okay, these are there. However in other case these are there, okay. So when antibody is diffusing, so here, okay, let's see, lots and lots are there, and this one also. So the reaction is happening in such a way that they make a line of precipitation, this one, these two, because there is a one set of antigen which is coming towards one side and this is another set of antigen which is coming towards another side, okay. So so what is happening, this is making one line of precipitation with antibody, another one is making uh, this line of precipitation, here the optimum concentration of the antibody meet, okay. Or in another scenario, and also it depends on the antigen concentration also, okay. So there what you can say, so suppose at this junction it got the optimum concentration between antigen and antibody, but here it may be at little lower or a little further, so that is why also there is a difference in reaching the optimum concentration, that is why they both cross each other, okay. But if that is a similar antigen, okay, so suppose from this one, okay, and if they will start coming in a single way, so suppose here, and if they both are same, they'll mix with each other, right, they'll mix with each other, and then the common concentration where they both will uh, like all will occupy the antibody. Okay, that is the thing we are asking now. In this case, what is happening, they both are making precipitation or the line of precipitation when all the antibody sites they are occupied with the antigen. Okay, so that is a common thing, like all the antibody is saturated with the antigen. So if they both are same antigen, they'll also diffuse in such a way that they mix with each other. They mix with each other, and then the line will be in such a way that yes, from both the sides the antigens are coming and they are same, and now they are interacting with each other. They have also mixed with each other, they are interacting with each other, and that is why they'll make an arc. It will never go like this. Okay, from this one it is coming and let me make a line where this will differ from this. That will not happen. If it is different, that means that here some other antigen is present which is interacting here. It makes an arc because they both mix with each other. Okay, lines fuse smoothly, forming a single and continuous arc. That means they both are mixed and making a single arc because they both altogether have reached an optimum concentration where the antibodies are occupied with this particular epitope. That is why they are making an arc rather than a cross. Is it clear?
Yes, ma'am.
Okay, okay. Okay, let's move ahead. Another technique is this western blot technique. Okay, so here also, in order to do the western blot, you need to do uh, first you have the antigen with you. Okay, so um, okay, at the end result is, or at the end what you want to do, you'll have a serum. Okay, so if I'm an infected person, I'll give my blood, and from that blood the doctor will collect the serum and try to recognize whether that serum contains the antibodies or not. And he has the antigens, like suppose antigen 1 for one particular disease, antigen 2 that is specific for another disease, and he'll use my serum and see my serum binds with which particular antigen, antigen 1 or antigen 2. Okay, and if it binds to antigen 1, means I am infected with this particular, the or the organism which produces this antigen. Okay, and if and it should, and it is usually if interacting with first antigen and it is not interacting with second antigen, that means I'm infected with the organism which produces antigen 1, not with the other disease. So suppose my serum and has antibodies which can react with plasmodium antigen, that means I'm having malaria, and it is not interacting with antigen which is leishmania. From the leishmania, I'm not having leishmaniasis, I'm having malaria. So that is a kind of uh, what to say, diagnosis method. Okay, and how much antigen or antibodies are there. Okay, like suppose I'm highly infected, I'll have highly or a lot of amounts of antibodies, or if it is second infection, I will have lot of amounts of antibodies in me. So small amount of serum is enough to able to recognize that. Okay, so that also the titer of antibody, that we say titer of antibody. So if the antibodies are less, then I have to give a lot of serum in order to identify that much antigen. Okay, so this is what we call a titer of antibody, that also we can detect. I'll talk about that in the next technique, but this technique is like just an detection, used as a detection technique. So it is called as western blotting technique. Okay, so there are southern blot, northern blot, western blot, all these three kinds of blot. Okay, so northern blot is for the detection of RNAs. Okay, southern blot is for the detection of DNA, and western blot is detection of proteins. Okay, so here we are using or want to analyze the antigen antibody interaction. Antigen is a protein, and for the detection of protein we are using this technique which is called as western blot technique. Okay, so okay, so first thing, we need to have the antigen with us, so that I explained previously also. So are you clear how we get the antigen which we want to be detected by the antibody? Is that part clear, or shall I explain again? The antigen part,
Clear, ma'am.
that is clear. Okay, so here also, uh, so once you get the antigen, either what you'll have a purified protein or you'll have a mixture of proteins. Okay, you have a mixture of proteins which you have to do the separation. Okay, and in this separation we use this SDS page, okay, which is a sodium dodecyl sulfate page, polyacrylamide gel electrophoresis. It is a gel electrophoresis. Okay, so this is an SDS page. So what it does, when you load your samples, if it has a mix of proteins, it will separate all those protein according to their size, higher to lower size. Okay, and this has having, and for the detection of the size we load a marker. Okay, a marker specific for each size. Okay, so suppose in your marker you know that okay, this is a marker which detects the protein of 25 kilodalton, then above that you have a 35 kilodalton. So if your particular you want to detect the protein or the bacterial protein or whatever protein or parasite protein at 25 kilodalton, so whatever signal you are getting, you will see whether it is coming near 25 kilodalton or not. So that is why a marker also we run with our samples. So first step is the separation. Okay, so first step for the western blot, we run the samples on a SDS page. Okay, or extracted proteins from the cells or from the tissues, we run them, and then they separate according to their size. So the next step will be, this is in a gel form, these proteins are on a gel. Next step, you transfer those from a gel to a membrane. Okay, so this membrane is called as nitrocellulose or PVDF membrane, where the protein samples can be transferred. Okay, so this is a transfer, you can see. So here it is a kind of a sandwich method. Proteins are having this charge. Okay, and then you put this is a sponge, this is a gel. On top of gel you are having a PVDF membrane, and there is a filter paper, and then there is a sponges. So it is kind of a sandwich. Okay, so your protein is majorly negatively charged coated. Negative coating is there on your proteins. So in order to transfer these negatively charged proteins to a membrane, what you do, you run or perform this, put the electrode here where is a positive charge, and then the transfer will happen. So the proteins will move from negative charge towards the positive charge, and then they'll be transferred to the PVDF membrane. Okay, so now the samples are moved from gel to this PVDF membrane through this transfer method. Okay, so after the transfer, what happens, then you'll get the protein transferred on this particular membrane, which is called PVDF membrane. Now this membrane is having all kind of proteins. Then in you block this membrane. The first step is a blocking step. Okay, so that membrane you take out, put that on a box, and then add milk or BSA. Okay, so the purpose of this is they are considered a blocking agent to prevent the non-specific binding. So there are several proteins which are having unsaturated sites. So this BSA, which is a common protein, okay, at particular concentration it acts as a blocking reagent. So it will block those non-specific binding sites and allow your particular or the efficient binding of your particular protein of interest with the antibody. Okay, so the blocking is a very crucial step, otherwise you will get lots and lots of binding which is could be non-specific. So to prevent the non-specific binding, you do the blocking. After you transfer all the proteins on the membrane. Okay, then after the blocking is done, you allow your serum to to bind with this. Okay, so remove the blocking, then add your serum which is supposed to have the antibodies. Okay, so if it is a specific antibody, if your serum contains the antibody, you have on the membrane you have the antigen present. Okay, the antibody go and bind to that particular antigen. Okay, now the binding is done. If it is a positive signal, the binding is done. Next step, now you want to detect. You cannot detect like that, like on paper you cannot see what is happening on the membrane. So then the second step will be secondary antibody you give. So suppose, okay, so this serum, okay, you have taken the serum you have taken from me, like a human. Okay, the secondary antibody will be anti-human antibody you will give. Okay, where these antigens or antibodies are raised. Okay, there you will have the human IgG, so the secondary antibody will be anti-human. So there those secondary antibody will bind, so this is a antigen with which the primary antibody is bound. Then there is a secondary antibody you will give which will bind to this particular primary antibody, and this secondary antibody is usually linked with the signal molecule, okay, or a enzyme that will produce the signal. So there is an enzyme, okay, and usually it is a horse radish peroxidase. Okay, it is a peroxidase. And when so this is done, so now you have added a secondary antibody also. Its secondary antibody is tagged with a enzyme which is just a HRP enzyme. Okay, so then now you want to see whether your signal is coming or not. So that is detection step that you do with the help of a substrate. Okay, so you give this enzyme its substrate. So now on the membrane you add the substrate of the antibody, and this antibody will react with that substrate and produce a signal. Okay, so for example in HRP there is a chemiluminescent solution along with H2O2 that you give. So this enzyme will work on that substrate, and on the X-rays it will give you a signal. And when you will visualize on your screen, now with the help of this signal you will be able to detect these bands. Okay, so you have run your entire sample, there were lots and lots of proteins, you did the blocking, so the non-specific sites are blocked. After that you have given the primary antibody with which you have given the secondary antibody which is conjugated with an enzyme. Then on top of this you will add the solution of your substrate, and then there the reaction is happening which you are able to see on the screen and after the chemiluminescent development. Okay, and this you can see in a form of a band on a paper. You can see on form of a band you are able to get a signal, and there is a molecular weight marker. Okay, so it is coming near 50 kilodalton. Okay, 50 above the 37, and whatever antigen you have given or the protein that you have produced, it is of the same size. That means this antibody is interacting with that protein, it is giving a signal. So that is what you aim for. So this is your entire Western technique which is you use to recognize an antigen on the PVDF membrane. Okay, is that clear? Clear, or you have any doubt?
No, ma'am.
No? Okay. If you have anything, any confusion, you can ask me, you can stop me and ask me. Okay. Similarly, we have ELISA. Okay, so ELISA is enzyme linked immunosorbent assay. Okay, so it is the principle is it uses enzyme labeled antibodies. Okay, enzyme labeled antibodies, the same concept to detect the antigen or antibody. Okay, so here you can see there are these types of ELISA. There is a direct ELISA. Direct ELISA means, so this ELISA we usually do in a 96 well plate. Okay, so earlier I have talked about the titer of antibody, right? How much antibody is present. So for that what we do in a 96 well plate. Okay, so you know in culture we have certain plates, so which is having wells in them. Okay, so different wells. Okay, and then you give antigen or you put antigen in these wells in increasing concentration. Okay, so small concentration, then more, and then more, and then more. Okay, and then you look for the signal. Similar to the Western blot technique, you look for the signal, and suppose at the lower concentration also if you are able to get the signal, that means your antibody titer is very high. Okay, and like lots of antibodies are present which can even detect a small amount of antigen. Okay, so this is what you can do. So there is a direct ELISA. So in this well, okay, these are wells where you can coat these wells with antigen. Here you can see this antigen coated well. It is a cross link. You can link with the or coat with the antigen these wells, and then put the antibody or the serum. If the serum is having antibody against that antigen, it will bind to that, and this is usually linked with the enzyme. Okay, it is usually linked with the enzyme, so it is a specific thing. So it is, these antibodies are generated separately, or lab generated antibodies, because it is having conjugated enzyme with it. So it is a direct ELISA. Antigen, antibody, directly you are able to see the signal. Then there is an indirect ELISA. You have the antigen, then this is a serum, so this is a serum antibody you are giving. Then there, to detect that serum antibody, you are giving suppose anti-human secondary antibody, which is linked with the signal giving enzyme, and then there is called as indirect ELISA. Then there is a sandwich ELISA. So between two antibodies you are making a sandwich with the antigen. So this is antibody coated, then you add the antigen, this antibody captures this antigen, and then you give the primary and secondary antibody. It is a sandwich ELISA. Then there is a competitive ELISA. Okay, so you give two kinds of you have an inhibitor and of antigen, and then you have this particular antigen, and then you'll see with the competition which one is able to bind. Okay, so there are these kinds. Okay, so they help in the diagnosis of the infection, for example HIV, COVID-19, measuring the hormones and cytokines, drug testing, and everything. Okay, so this is also showing the same thing. Direct may you will see this antigen, then primary antibody itself is linked with enzyme, and then when you give the substrate it will produce a kind of color. So in this 96 well plate you will able to see the color, and then intensity of that color you can measure with the plate reader, that will give you how much intensity it is having. Okay, then also this is indirect ELISA, antigen, primary antibody, secondary antibody, and this is the sandwich ELISA. Then this is what it is. Okay, this is also clear, similar principle. Only thing here you are able to check the titer also, because lots of wells you can give in a single time. You can just load different concentration, you will measure the signal in the terms of color intensity. You can measure the titer of the antibody, at which concentration it is able to bind, at which lowest concentration, and also titer you will be able to know. Okay, this is also same principle. Then you have this FACS, which is fluorescence activated cell sorting method. So this is also a specialized type of cell cytometry. It not only analyzes the cell but also sorts them, but based on specific properties. So very useful in immunology, cancer biology, and all these techniques. So here what you have is cells are labeled with fluorescent antibodies. Okay, so these antibodies are tagged to specific protein or markers. These cells are passed in a fluid system stream through a laser beam, and detectors capture the scattered light on the basis of size and granularity and the fluorescence emission. So based on these signal, cells are identified, counted, and can be physically separated. For example, this one. Okay, sample strained in a cell suspension, they are streamed through this. Okay, sheet fluid. Okay, and then there are certain proteins which are tagged with a specific marker. So here you can see the detection laser light source, which helps in detection of forward and side scattered light from all cells, and detection of fluorescence emitted by stained cells. Okay, so which cell will take the stain, that will give a different kind of fluorescence. Okay, this is also the same thing. These are different filters, these are different detectors, then amplification, and then the conversion to that in a signal. Okay, so let's see. Let's go through the question answer. So on a radial immunodiffusion assay, the area of precipitin ring is proportional to. Okay, the first graph that I have shown, it is proportional to what?
Concentration of antigen.
Yes, concentration of antigen. The more the antigen, the bigger the area of that radial precipitin ring. So then an exogenous protein was overexpressed by a student in some cell lines. So which technique would be the best to determine the overexpression of this protein in a cell lysate? Okay, so
West, Western blot.
yes, western blot. And what do you detect with southern and northern blot? With southern blot you can detect?
Ma'am, northern blot is for RNA and southern blot for DNA.
Yes, good. Yeah. So yeah, western blot is the correct option. Then in the immunostaining technique, paraformaldehyde is used. Okay, so this is a fixing agent. Okay, so suppose you want to, okay, so in a slide you put your samples, okay, you put your cells, okay, and then you want to visualize them, or just simple ELISA or something. Okay, so on a glass slide, why cell will stick to that? Okay, so that fixation on the slide of that particular cell, you do use a fixating agent. Okay, fixing agent, fixative you use. So that is usually of paraformaldehyde. It crosses with the membrane and allow them to stick to that particular platform where it is present. Okay, so that helps, or it is fixing agent. Okay, so this, there are several steps. So this is immunostaining. So immunostaining is also, so suppose I am having a cell line. Okay, I'll spread them on the glass slide. With the fixing agent, I'll allow it to be fixed on that slide. Okay, it remains fixed on that slide. Then also the same step. Okay, blocking you will do. Okay, after blocking you'll also do a permeabilization, as those cells are very intact, they are intact. So you'll use a permeabilization agent. Okay, so you, that permeabilization agent will make some pores, or it will permeabilize the cell in such a way that it allows the antibodies to enter inside the cell, because you have a cell which is surrounded by a cell wall, and inside that cell you have the protein, and then you are suppose putting the antibody, so it is like entirely covered with the cell wall, right, from all the surroundings. Then you put the antibody. So if it is covered by the cell wall, how the antibodies will enter inside? So for that purpose you'll have one step which is permeabilization agent. For other of our techniques we were not having this permeabilization step, because those you are lysing the cell. Okay, you are breaking the cell wall of the cell and then loading on the SDS page, or on it is a purified antigen that you are coating on ELISA plate. Okay, so that is, that antigen is directly exposed to the antibody. Okay, it is ready to expose. But here in this case, when you are giving a full cell, and there you want to recognize whether a protein is present inside this cell at this moment or not, so there you have to do this permeabilization step, allow the pores to open in a small, like a limited amount, so that the antibody can enter inside the cell. So that is a permeabilization agent. So we usually use Triton X or saponin. So these are some permeabilization, or a kind of detergents, harsh detergent, which will break the cell wall and in a limited concentration, and then the antibodies can enter inside the cell. Okay, so blocking, permeabilization, then again you give this primary antibody which can enter due to permeabilization, then again secondary antibody, and then the detection. Okay, so if it is a fluorescence technique, you put on a wavelength of certain, like suppose you want to see the green fluorescence, you set up a wavelength, give a light of certain wavelength that will produce green color. So if the antibody has cross reacted with that antigen, the reacted with that antigen, and the secondary antibodies is able to, it is conjugated with the fluorophore. So if all these steps have been done, when you emit, or like give the light of that particular wavelength, it will emit the fluorescence. Okay, so that is what you can detect. It is a fluorescence technique with the help of, it is called immunostaining. Okay, with the antigen, with the help of antibody, you can detect, stain the cells which are having those antigens. But suppose if at that particular moment your cell is not expressing that protein, your antibody is going inside the cell, but if the protein is missing, or it is at a very low concentration, it will not able to react, it will not able to react. And after, so there are also, you are having a washing step. So whatever unbound proteins or antibodies are there, with the washing step all those antibodies will be removed. If the protein is there, an antibody has bound to that protein, it will remain with, attached to that antigen. With the washing it won't come off. But if the antibody has gone inside and protein is not there, antigen is not there, it is not able to bind, then when you do the washing, the antibodies will come off. Okay, so that is the entire technique. Clear? Is it clear?
Yes, ma'am.
Okay. Okay, what is the purpose of blocking in ELISA? Okay, so to promote binding of antigen, to enhance the sensitivity of the assay, to inhibit the activity of enzyme, or to prevent the non-specific binding of antibodies.
To prevent non-specific binding.
Yes. Yeah. Okay, certain statements are made below regarding ELISA. Okay, let's see. Which one is true? I guess we have to say. We have to. Okay, the first is it is highly sensitive technique for detecting and quantifying soluble substances such as proteins, peptides, antibodies, and hormones. Okay, it is a sensitive technique used for detection and also for quantification. Right? We can quantify, right? Like suppose you are doing an this one, this ELISA. Suppose you have coated the plate with the antibodies, you can say. Okay, and then you give each well having same, coated with the same thing, and then you will give antigen. Okay, similar amount. So there, if there is a different serum sample in each one, okay, and all are having different uh, amount of antibodies, so whatever antigen, if the antigen concentration you are keeping the same and then allowing the detection, so one well or from the serum from one patient, if it is having more antibodies with the same antigen, it will give like that much antigen, it will give more. Okay, because you are giving enough, it will give more signal. Another one which is having lesser antibodies, it will give less signal because unbound antigen will come off, so less signal intensity. So this is you can use for quantification. Okay, so first one is correct. Quantification you can do, which is a highly sensitive technique. And then is it uses antibodies labeled with enzyme that produces a measurable product when substrate is added. Correct? Yeah. Then is the C is the technique does not require separation of free and bound antigen. Yes, and then the color change in ELISA is indicates presence of target analyte. Yes, yeah, the color change shows the presence of the particular analyte. It is possible to use monoclonal antibodies in. It is possible to use monoclonal antibodies in ELISA. Hmm. And the yeah, you can use. And then the intensity of color produces directly proportional to concentration of target analyte. Correct? Yes, yes, yes. E is incorrect. F is correct. Yeah. And this one D also. So, and so the correct sequence for Western blotting. Okay, so what is the sequence for Western blot? Performing the polychromatic electrophoresis. Okay, this is, and then transfer proteins by the to the
Ma'am,
PVDF.
option C.
Okay, what is option C? First is proteins are denatured by STS and by boiling incubate PVDF. So first step we will do this one. Okay, the first step is to prepare the sample. Okay, so you have the cell, lyse that cell from the with the help of SDS, boil it so that proteins become linear. Okay, this is the first step. Your samples are ready. Now after the samples are ready, you will run them on a polyacrylamide gelato at this page. Okay, for the separation. Now the separation has happened, you will transfer the proteins to PVDF membrane. Transfer step will happen from gel to membrane, and then incubate the PVDF membrane to primary antibody, secondary antibody subsequently, and then detect the signal. Okay, correct. So this is the third option. Okay. In the immunostaining technique, the proteins are stained with which one? Antigen? No. Um, so proteins are stained with protein itself they are using, so it cannot be stained with antigen, it cannot stain with comacillin glue. Okay, silver stain also not, zinc stain also not, antibody conjugated the fluorophore, right? Yes. So in the flow cytometry, what does this forward side scatter measure? Sorry.
Cell size.
Okay, yeah. Okay, so facts, the cells are sorted by cells as electrostatic force, magnetic force, dilation, plating until they are only single reactions and differential board. Yes, electrostatic force. Yep. Okay, so in the immunodiffusion test, if two lines cross each other, then what happens? That means what? Two antigens are identical, not identical, partially identical. What?
Not identical.
Yes. They are not identical, that's why they cross. Otherwise, they'll make an arc. Okay, that's it. These are the questions for today. You want to ask anything?
No, ma'am.
Okay, if there is no question, we'll conclude the session today. Okay, thank you for joining.
Thank you, ma'am.